All right, everyone, we're going to get started with day 3. This is our session where we're going to be seeing what people have been up to in other aspects of spatial analysis, seeing which ideas we can steal for our own nefarious purposes. So starting us off is going to be Lauren Gillespie, who's going to tell us about citizen science. Lauren, go ahead.
Sorry, figuring out which mic to use. Hey everyone, so I'm Lauren. Thanks for the introduction, Noah, and I'm a rising 5th-year computer science PhD student here at Stanford. And so yeah, today I'll be talking about how we can use deep learning, citizen science data, and remote sensing to map plant communities across space and time. So previously in this workshop, you've learned a lot about spatial biology, but at a very micro scale, right? We're talking about cells within tissues, within organs, as you can see here from Noah's paper in Nature Biotechnology. But today we will be talking about a much different scale, in fact, 6 orders of magnitude bigger, to the scale of ecosystems. So within this, from my perspective, what I'm looking at is species embedded within communities embedded within ecosystems as compared to cells embedded within tissues embedded within organs. And while they may seem like very different things that we're talking about, while the scales are different, they're actually quite similar. For example, many of the methods that I'll be showing today are very applicable and have been brought up in other talks during this workshop, including machine learning and GIS. And again, if we think about, even though the scales are different, the kinds of organisms that we're looking at, there's some nice parallels. So as I mentioned, you've seen a lot of going from the individual cell level to the tissue to larger organ systems. And in my work, I go from individual species of plants to local communities made up of multiple species to entire continent level ecosystems. And so why do we want to talk about plants today? Well, that's because plant biodiversity is in peril, as you may know. So there are many ongoing factors affecting plant biodiversity around the planet, but one of the most strongest that you've probably heard a lot about is deforestation and what we call more broadly land use change. So this is Rondônia in the Amazon over a 30-year period, and you can see that much of the forest has been removed to clear the way for soybean growth. And so this is changing ecosystems so rapidly at a scale that hasn't been seen in time immemorial by these ecosystems. So this is change that's happening way outside of the distribution of what these ecosystems are used to. But this isn't the only kind of change happening. There's also climate change ongoing, specifically climate warming. So this is the Columbia Glacier in Alaska. And as you can probably see in the center, the glacier has retreated a lot just in the past 30 years. But I also want to point out down here, clicker's not going to work. Oh, well, in the bottom right corner, you can also see that it's starting to green as well. So this is this Arctic greening effect we've seen as the climate is warming. Plant ranges are shifting in response to this climate change as well. So it's not just land use change, but also the climate as well. Multiple factors that are affecting how plant biodiversity is changing around the world, thanks to humans. Now, the problem, though, is that traditional methods that we use to track biodiversity and its change just sort of lack the resolution we need to actually detect most of this ongoing change. So what I'm showing you here, this is a plot. So this is what traditionally ecologists have used to track biodiversity change over time. They'll go take a bunch of stakes and stick them in the ground, and then they'll come back to them repeatedly over years. And so as you can see, this is the evolution of a plot in a marshland ecosystem over time. And you can see that it's a little bit hard to tell, but the plants that are in this ecosystem have changed dramatically with saltwater incursion. And what's great is that these sort of plot-based monitoring networks, as we call them, have really grown in scale over the past 30 years. And so this is a map of one of the largest distribution or largest consortium of these plots across the world. And as you can see, we're almost to the point where we have multiple plots in multiple countries across almost every major ecosystem. But as you can also see, we have 10 plots in all of California. And California has 10 separate unique ecosystems. So we're talking about one plot per ecosystem per 500 kilometer square resolution. And that's just not enough scale for us to track these sort of fine-grained changes that we were seeing from, say, land use change over time. Similarly, other methods that people tend to use for tracking biodiversity change are what are called species distribution models. And so what that means is what we're going to do is take rather than going and looking at the same place over time, we're going to just opportunistically collect a bunch of observations of a given species across some landscapes. So we're going to go and we're going to like take our phone, we're going to go look for a redwood. And whenever we find a redwood, we're going to maybe take a picture of it and we're going to mark the GIS location of where we found that redwood. And what we're going to do is we're going to take an environmental variable like what you see in the top right. This is a climate raster. And we're going to try and associate those known observations with this underlying environmental variable. And then we can build a model that we can then project or interpolate across an entire landscape to get an estimated range of where we think this species does and does not grow. And while this method is great for detecting large-scale biodiversity change across thousands of species across the centuries timescale and can help us detect where industrialization, for example, has affected plant ranges most strongly, as you can see from this example, though, these climate rasters are incredibly low resolutions for the kinds of changes that we're interested in my work. So as you can see in the middle, this is remote sensing data for the exact same area as the climate rasters. And then below this is the local community composition. So each of the individual unique colors is a different plant community. And as you can see, our climate rasters just don't capture that variation that we need in order to detect local communities. And so it's able to capture rough changes in space and time, but at this fine granular scale that we're interested in, it just doesn't have the information or the resolution. So that really brings us to the point of my work, which is that we really need these higher spatial and resolution methods for detecting plant biodiversity change over time. And the things that we have so far, the methods we've developed so far, just aren't cutting it. Now, for my work, the kinds of data that allow us to start to answer these questions are remote sensing data. It has the spatiotemporal resolution that we need. So here on the left, you can see these are the individual tops of old growth redwoods in Northern California. You can actually see the individual trees. And on the right, we can see here over a 2-year time span, we can a wetlands restoration project in Northeastern California. And so compared to the climate rasters that I showed you 2 slides ago, this sort of data gives you a resolution that's just uncomparable to the climate data that's usually used to detect biodiversity change. Similarly, citizen science, which is normal people going out and trying to collect biodiversity data for the purposes of research, have been really expanding in recent years. Some of y'all may have heard of this app called iNaturalist, and what you do, it's kind of like social media for nerds. You like go out and you take pictures of animals and plants and fungi, and then you can upload them, and then people can help you identify them. So like if you don't know what kind of species of fungus is, this will help you to determine what that is. And what species that is, you'll get the rest of the community to help. But what's nice though is that with iPhones or phones in general having such high GIS precision, those lat longs of where you observe that species can also get uploaded as well. And then scientists like me now have a bunch, like literally over 1 billion records of where certain species have been seen at certain times and in certain places, which is just a trove of information that we haven't had recently up until like the past 3 or 4 years in terms of like this density and this quality of individual species level information. So this is the web of all of the observations we have across the planet, which is pretty crazy. So the bulk of my work aims at how we can build models to use this remote sensing and citizen science data. And the technique that I use to do this is deep learning. Coming from a computer science background, that's sort of my specialty. And so now I'll show you how we tried to do this for this plant biodiversity mapping problem. So the first thing we did was we built a machine learning optimized biodiversity prediction data set. So on the left, you can see these are those citizen science records from across the state that I mentioned. So each one of these little dots is a picture that someone has taken of a plant and uploaded it to the iNaturalist website. And then the color is just the density of observations in that area, since there's too many to plot without breaking the computer, screen. And then in the middle, what we do is we take each of those individual dots and then we pair it with a remote sensing image cropped to the local area around where that observation was taken, specifically a 256 by 256 meter or pixel image around that location, which you can see here in the middle, some examples of from across the state. We also take that low resolution climate data as well, because we also know that climate is very important for plant ranges, as I showed you in the beginning of the presentation. So these are our 3 streams of data that we have for this modeling task. Now, as you probably are wondering, we have 2 very different resolutions with the data we're working with, right? This remote sensing data is at 1 meter per pixel, but our climate data is 1 kilometer per pixel. So how do we deal with this mismatch in resolution? So for this project, we went with a very simple ensembling approach. So we have one, what we call head, that is a convolutional neural network that's very good at detecting 2D patterns in data. So that's what you see here on the bottom. So that's taking in the remote sensing data. And then on the top, we have a simple, what's called multi-layered perceptron, which is like a very standard neural, like vanilla neural network, which then takes in those low resolution 19 climate variables at a given location. And so ensembling those predictions together, the model then predicts not just species, but also genus and family, because some species are rarer than others. And so by including higher taxon information, this helps the model to improve its predictive capabilities for those species that it maybe doesn't see a lot of observations for, but they have, that species has a similar individual in its genus that there are a lot of observations for. And if they share a similar environment, then that can be helpful for the model to learn how that one species, where that one species lives. So with this deep learning approach, what we see is that it does indeed outperform all of our other previous approaches, such as other remote sensing based approaches and deep neural networks, which is this inception model you see here, and also climate only based deep learning approaches, which is MLP, multi-layer perceptron in yellow, but also more classic species distribution modeling approaches, which I showed you in the beginning, which are what traditionally ecologists will use to track species and their changes. That's maximum entropy methods, and then also random forest as well. And so we see an improvement of a performance improvement across a wide variety of accuracy metrics. We actually looked at over 20, but I'm only going to show you 3 today because I don't want to bore you with too many statistics. But hopefully this proves that the model does indeed improve species presence prediction. So on unseen observations of species, it's able to predict them with a higher accuracy than the other approaches, which is great. But how do we actually like operationalize this? How do we use a model that can predict species well to track biodiversity and its change over time? So that's the next section I'll be talking about, how we actually track biodiversity across space. So I'll start with a little case study here in the Northern California in the old growth redwoods. So hopefully you can see that there's sort of this like olive band running down the right-hand side of this image. And so these are old growth redwood groves that are remaining post clear cutting in the 20th century. So 95% of redwoods were old growth redwood groves were clear cut in the 20th century, leaving just a few pockets of these old growth groves, which are actually very easy to see from satellite imagery. So easy, in fact, that humans can annotate them directly from satellite imagery, which is what you're seeing here on the left. This is human annotators detecting essentially where these old growth groves are remaining in this landscape. And then as we can see on the right, this is the official National Park Service map for this region. And the red regions are where old growth redwoods are. And as we can see, they map quite nicely to one another. Now, I won't explain how we go from species predictions to mapping. I'm happy to explain that during the Q&A session if people are interested. But here is the same map, but using deep biosphere, using this neural network architecture to make predictions across space for specifically redwood species. And what you can see is that it predicts a much larger range, a much larger extent than what the humans predicted. But if you look again at this map from the National Park Service, we see that there's actually 2 types of redwoods. There's not just the old growth redwoods, which are in red, there's also secondary regrowth. So these groves that are regrowing after being clear cut in the 20th century, which you can see in blue. And indeed, it does look like our model, Deep Biosphere, is capturing both kinds of redwoods quite nicely. And in fact, if we go and we take each pixel and we plot whether or not it's coded as a redwood using the official National Park Service map, we do indeed see that our model is the only one that on average predicts redwoods as present in pixels that are coded as redwood by the National Park Service, which is pretty exciting. This is again compared to human, another deep learning remote sensing based approach, and also maximum entropy, which is a more classic species distribution modeling approach. So this is great. But again, humans can also see these redwoods. So what's the special kick for using a neural network? Well, I think I forgot to mention in my my slide, but we're making predictions for over 2,000 plant species simultaneously. And that includes species that are very small, species you can't directly observe from remote sensing imagery, such as redwood sorrel, aka Oxalis oregana, which you can see here on the bottom left. And so this is a small ephemeral under canopy species that only grows under other trees rather than out in the open. So you'll never be able to directly see it from remote sensing imagery. But what we do know from the National Park Service surveys is that Oxalis oregana prefers to grow under old growth redwoods compared to secondary growth redwoods. And so what we can see here is that at the predicted range from Deep Biosphere for Oxalis oregana, looks like it is predicted most strongly in areas that more or less vaguely correspond with where the where the old growth redwoods that I showed you earlier are. Conversely, this is Douglas iris, Iris douglassiana, which is another under canopy species that has a preference for young secondary regrowth redwoods. And we see this opposite effect where we see it has a higher predicted presence in areas that are regrowth. So forests that are returning, but maybe not as full and thick of a canopy is what you see with the old growth. And in fact, if we take these two predictions from these two separate understory species, and we take the difference of those predictions, we do indeed see that Oxalis oregana redwood sorrel is predicted more likely to be present compared to Douglas iris in places that are associated with the old growth redwoods, or pixels specifically, as compared to, and vice versa for Douglas iris, which is really great and shows that the model, even if it's not able to directly detect the species, is also learning species interactions as well. So learning that old growth redwoods tend to be a better environment for redwood sorrel and vice versa for Douglas iris and secondary redwood growth, which is really awesome because many of these species, again, we cannot directly detect from remote sensing imagery, but if we can link them to things indirectly that we can observe, then that's very exciting for unlocking the potential of remote sensing for ecological questions. So the last thing that I'll talk about is how we can actually map biodiversity across time. This is sort of the holy grail of biodiversity monitoring. It's great that we can track biodiversity across space, but time, that change that I showed you at the very beginning is what we really care about. So this is another case study. This is in the foothills of the Sierras, just east of here in California. So this is the Rim Fire, which happened in 2013. And you can probably see that the burn scar is quite very visible on satellite imagery. And in fact, if we look at the empirical burn severity metric, so this is how hard the fire burned in a given area, or strong, sorry, you can see that indeed there are many places where there was extreme fire behavior and subsequent scarring. So if we then take our model, our deep biosphere model, and we take predictions for species using imagery from 2012 and compare it, which was before the fire, to imagery taken in 2014, which was after the fire, and we take the difference between these predictions, how much have the predictions changed, then we get this map. And as you can see, visually speaking, that it does indeed appear that deep biosphere is detecting larger changes in places that are more severely burned. And in fact, we have the statistics to show that is the case. And it's not just that it's naively detecting where the remote sensing data has changed the most, because if you do the same comparison with just the raw remote sensing data, there's not near as much signal as what the neural network is predicting in terms of change. So that's really exciting in terms of ways that we can get towards this change detection more easily than what we have traditionally done with just raw remote sensing alone. And so with that, I would like to thank y'all for listening. And unfortunately, I won't be able to stick around for Q&A because my summer intern is giving his big talk. But if you're interested in talking more or have any questions, please do reach out. My contact information is at the QR code. And with that, I'd love to take any questions.
Was awesome. Thanks, Lauren. So anyone who has questions, please walk up to the mic.
Yes, very interesting. I was wondering about your modeling prediction, where you said something that you use if you have a lot of data from one species within the same genus, then you extrapolate that to other species within the same genus. And I was just wondering whether you can actually do that. If we take something like the finches of Galapagos, which are within the same genus, but they are very different, they live very different, they eat different things. So I don't know whether, so I'm just wondering, have you thought about what you're, like you're missing out on something, or you're predicting wrong, basically?
Yeah, so that's a good point that taxonomic space is not necessary, or taxonomic similarity does not directly imply like environmental similarity, but there is a strong correlation, at least with plants, because oftentimes plants in the same genus or the same family will have very similar traits. And to have the same trait, you usually have to be in very similar environments for those traits to be useful. But it is true that there were certainly cases where that's not the case, where you'll have one plant that lives in one ecosystem and then the other plant in the same genus that lives in a different one. But there's this shared evolutionary history, right? And so oftentimes that's baked in as having similar traits or occupying similar, say, niches. So like, for example, with redwoods and sequoias, they don't live in the same area, but they they have the same growth patterns, like their canopies look very similar. And so, and even though they're in very separate ecosystems, that shared evolutionary history oftentimes will still provide some signal for which the model can pick up. But that is a good point. We still also include the individual species information, so there'll still be signal about like when you have two individuals that like the genus is the same but the species is different, so you'll have those predictions in separate areas. But that is a good point. Like something we want to work on more is how to bake in others, like metadata essentially that we have on like where we expect certain species to be and not. So that's a good point. Thank you.
Thank you.
Hey, great talk. So I was just wondering, in immunology and in microbiology, we're using a lot of these tools to help inform like diagnosis or staging of different diseases. And I'm curious like how this deep learning model is being used to inform like policy and how the findings differ from other sources, and kind of where do you see that leading in terms of like impacting how we approach climate change?
Yeah, that's a great question and something that we're very interested in. But when we're talking about policy, like oftentimes you need you need to be very certain essentially. And there's this issue that like deep learning is not interpretable. So how can you trust the predictions, which is very valid. And so right now we've been mainly operating in the academic sphere rather than working in the policy world. But that's something that I've been keeping in the back of my mind because it is, of course, like the ultimate end goal, right? And I think that what will be required, there's been some cool work actually out of the conservation biology community of sort of building like threshold-based models of where essentially policymakers can go and decide like what are different important factors for them for when they want to build, say, maps of like conservation priority, for example. And so I could definitely see a world in which you can sort of bake this sort of thresholding into these models. But I would say at the moment, frankly, that I don't think we can trust these predictions quite well enough yet to make policy off of them. But it's a step in the right direction towards that, right? And I think rather we're more going for instead of using for policy decision making, using it for some sort of like automated biodiversity monitoring, so that you can actually like put experts who have the most high quality data for making policy in the right places, is rather where I see this going than it being a direct, like, say, map of conservation priority. I think that's going to be a hard sell, given these models are not yet interpretable.
Great, thank you.
A question from online. For data analysis relying on citizen science, how do you account for sampling bias and, maybe people like hiking in certain areas more than others?
Yeah. I didn't have enough time to get into this, but this is a big problem with citizen science data. And in fact, that's why it's been very underused for a lot of traditional biodiversity monitoring methods. So for our approach, we do include there. We're not doing any like special subsampling, for example, based on what region or how dense the observations are. But how to calibrate essentially across for this heterogeneous sampling is something that we are thinking of for next steps. And I think that is why we see some performance improvement, or see the performance improvement that we do with the deep learning approaches compared to, say, classical species distribution modeling approaches, because patterns like these topographical patterns don't change as much as you can see here in this remote sensing data as they necessarily do with, say, climate data. And so when you have a bunch of observations in, say, like LA County, then your climate-based approaches will over-optimize to the climate of LA. But there's still patterns within the remote sensing data that might be useful for, say, like the Mediterranean ecosystem of the San Gabriel Mountains still looks quite similar to what we see in San Jose. So there's a bit more of a like signal for extrapolation there. But yeah, the citizen science data is very messy, and it's something that we've been working with. We did do a lot of data cleaning to make sure that we remove some of this over biased information. So like, say, a picture is taken of the same tree multiple times, trying to move that kind of information. But it's definitely a big open problem for the field, and definitely next steps for improving not just the prediction, but also, like I mentioned earlier, like the trustworthiness for downstream decision-making purposes.
Definitely. Are you factoring in other climate parameters like temperature, humidity, or these types of things into your model?
Yeah, so those bioclim variables that I mentioned, and those do have average or decadally average climate, temperature, precipitation. They're called bioclim variables, but they're essentially like climate variables that over the years ecologists have determined are like the most tend to best model plant ranges. So things like mean average precipitation of the wettest quarter, coldest month of the year, average temperature, stuff like that. So those are baked into those climate variables. The problem is that those are fixed in time. Like those are huge, like averaged across like 1970 to 2000. And so we're not getting that temporal resolution with climate. And so something I'm hoping to do at some point is maybe also include data from, say, MODIS or these other like weather satellites that NASA uses, kind of more real-time information for climate. But that data is much noisier than the climate data that you get when you average. So that's another added layer of complexity that we're trying to figure out how to account for.
Great. Well, please join me in thanking Lauren for a great talk. Next up, we have Sean, who's from SLAC, not the messaging app, but Stanford's SLAC, who will be telling us about the work he's doing with neural fields and differentiable simulation.
Yeah, thanks so much for having me. Yeah, I think it'll be kind of fun to have a completely different set of people for this. And so, yeah, like Noah said, postdoc at SLAC and the machine learning group, and not studying anything alive, just studying some stuff in atomic and particle physics. And so for some context on the actual signals and what we're interested in terms of the physics, then there are two kind of things that I want to highlight in this talk. One being this MAGIS 100 experiment, which is targeting gravitational waves, and then the other being DUNE, which is a neutrino experiment. And so both of these are physics experiments in development, and both have sort of lots of fun spatial signals that then we have some spatial analysis techniques. And like I said in the title, we're looking at some neural fields and differentiable simulation to analyze these spatial signals. And so a bit more particularly on the context. I'll have some high level questions associated with all these, but a couple of papers that I'd like to talk about today. One is on this idea of light field sensing and 3D reconstruction, little 3D GIF there. One is on some detector modeling in neutrino physics and also in this light field sensing context. And then the last thing is on signal and systematics extraction in atom interferometry. And so these are kind of very detailed things, but kind of the broad questions associated with them are for the light field sensing and 3D reconstruction, it's how do we infer 3D structure from 2D data? For the detector modeling, it's the question of how do we efficiently represent and calibrate models of our detectors. And so by calibration, I mean kind of match the simulations or the digital twins with real data. And then the last thing is how do we efficiently fit components of our data? And so these are kind of the big questions that I think are more broadly applicable than these contexts. So yeah, curious if they apply to spatial biology as well. On this first point, this 3D structure from 2D data, kind of the motivation here is that we've got this atom interferometry context. And so don't worry too much about the physics details, but basically we've got some underlying physical process, which is that thing on the left. And that results in some physical atom cloud, which is happening in 3D. So the 3D space is really where the physics is happening, where we have that. In practice, it's instead of measuring directly 3D, we measure these 2D camera images. And so, you know, there's kind of this question of, okay, if we're measuring stuff in 2D, but the actual physics is happening in 3D, can we kind of back things out, go back closer to where the physics is happening? And then ultimately, the idea is that maybe we can connect that back to the underlying physics. And so at SLAC, we developed this particular sensing apparatus motivated by this idea of, let's say, can we go from 2D data and back to something 3D? And the design principles here are maximizing the light collection, and we have some experimental constraints, so we want to do this in a single camera shot. And then also this multi-view imaging, which gets us able to do a 3D reconstruction of the atom cloud. And this concept is this dome of mirrors, which I have pictured up there. So the idea here is that you've got a bunch of mirrors in an array. Those are those circular colored dots there. And you have those pointing at a camera, so this lens sensor system. And if you suspend some object or some atom cloud in front of the dome of mirrors, which is that little black dot, then you get an image on your sensor from each one of these mirrors. And then you get a whole bunch of different views of your object or atom cloud that you're trying to image. And a bit more particularly, so we went ahead and built a proof of concept of this. So it's a 3D printed dome, and we're imaging little 3D printed cubes. The SLAC is a DOE lab. So the 3D printed little cube has D, O, and E on each of the faces. And to give it a sense of scale, this is about 1 millimeter for each side, this DOE cube. And like I said, you get an image on your sensor from each one of these mirrors. So each one of the bright dots in that big image there is coming from a different mirror. If you do some then image processing on that, just do patch extraction, then you get data that looks like this, where each of these images is a different view of the object of interest. And so then the kind of algorithmic question is, okay, we've got the data, how do we go to a 3D model? This kind of gets us to this idea of rendering an inverse rendering, which we spent a lot of time working on. So for rendering, then this is just from some 3D model or scene, simulating what an image on a camera is going to look like. Inverse rendering, the other way around, from 2D images, reconstruct a 3D model of the scene. So inverse rendering is really what we're trying to do. And the way we do this is this analysis by synthesis approach. So you start off with some representation of your scene or object with some associated parameters, and then you do your rendering procedure to get some synthetic data, and then compare that with your real data and try to match up, make your synthetic data match the real data as good as possible. And you do that by adjusting parameters of your scene or your object. And so in order to do this, you need both a renderer and a representation of your scene and object. The renderer, we built this ray tracing simulator at SLAC, and ray tracing in general, it's just a very efficient way of rendering a scene. And nice thing with a ray tracing approach is that you can do this with arbitrary optical setups, and it works pretty well. And this is just based on straight line paths of light. We built this also to be differentiable, which I'll talk about later, because we can do some fun things with that. And then in terms of how we represent our scene, how we represent our 3D objects, this idea of neural fields or neural radiance fields is one notable example. And neural fields in general are just continuous function representations of signals using neural networks. NERFs are a very notable example in rendering. If you've, if you're kind of in this space at all, you've probably heard of that. The kind of important thing is that the networks used for these NERFs are pretty simple. It's really just kind of the physics and the way that you use these that make them very powerful. And the way that these work is that you have some input images, and then you do this kind of rendering, this ray tracing procedure. And when you do this ray tracing procedure, what you're doing is integrating along array. And what that corresponds to in practice is that you have a bunch of 3D XYZ points along that ray, and then you also have an associated direction of that ray. And that spatial information gets passed into a neural network, and that neural network outputs some color and some density at each of those points that you're sampling. And then you can integrate along that ray, get a contribution on a given pixel, do that a bunch of times, and then do the procedure that I talked about a couple slides ago of this analysis by synthesis. So then this is just an example from the NERF paper of doing that. You can do a little moving around in 3D. For our context, what this looks like is you have this set of 2D images, do some training, get this nice GIF, and then you get a 3D model. And so again, it's this DOE cube. This is just showing kind of a level set at the surface of the object from a few different orientations. But these neural radiance fields or neural fields really are accomplishing this 2D to 3D task, which is great. And of course, in practice, we're not going to be doing this on these little 3D printed objects. We're looking at atom clouds. And so there's some work in progress, both with the Hogan group at Stanford of dome imaging of real clouds, but then you don't actually have to necessarily use our dome for this kind of multi-view imaging thing. And so we're working with a group at Northwestern to try to do this 3D reconstruction, but just using a set of cameras instead of this fancy sensing device. Yeah, and so that's kind of the first thing of how do you infer 3D structure from 2D data. But then kind of wrapped up in that was both we need some render, we need some modeling of our optical setup, and then we also need some representation of what our 3D object looks like. And turns out that this made all these GIFs, but turns out that this this neural fields as data representation can be very extendable and be very powerful, even beyond this kind of inverse rendering context. And so there's an example from neutrino physics, the paper linked there, where basically what's done in this neutrino simulation is that you do some very, very expensive simulation. You don't want to do it again. And so then you just store that as some voxelized spatial lookup table. So you have some binned up space of your detector that then you just draw from to get some simulation of a detector response. The problem is that neutrino detectors, like generally the sensitivity scales with volume. And so then if you want to have a big detector and keep a good resolution in your number of voxels, in your voxels, then this just scales really, really poorly. And so kind of the motivation is that we want to have something that scales a bit better, that doesn't require an explicit binning of the 3D space. And so the idea that we had was to just train one of these neural fields to represent this spatial lookup table. And yeah, it works fairly well. You see a little comparison between just training the lookup table is on the top there, and then the trained network is shown on the bottom, just a slice of the detector. But then, yeah, but these are nice because they're continuous functions, so no explicit binning, which means that they scale very nicely with volume. And then the other nice thing is that lookup tables, they're not very easily trainable, but the neural network is, and so we can tune this representation on some measured data. So basically just getting the point that these neural fields are pretty nice for representing spatial signals in a lot of ways. And then in the talk title, there's also this idea of differentiable simulation, and I mentioned a couple things of things being differentiable throughout the talk. And this, again, is this idea that 3D reconstruction and many physics analyses require detailed simulations of physics detectors. And in the 3D reconstruction thing, this can be like positions of cameras, mirrors, and parameters of the physical system for more generally. And if we can tune these digital twins to more closely match the data, this will improve our fidelity, either for physics analysis or for 3D reconstruction. And also, this is a bit broader. Kind of the ideas here, you can also fit some signal components, what have you, if you'd like. And a differentiable simulation is just a simulation where you can calculate gradients of outputs with respect to both inputs and parameters of that simulation. And you can sort of quote unquote easily do this by writing the simulator in an automatic differentiation framework. So this is something like PyTorch or JAX, these machine learning frameworks. I say easily because this is not always the most straightforward, but there are these nice tools out there. And the motivation for doing this is that if we want to do some fitting of things, gradient-based methods are very efficient, even when number of parameters in that we're trying to fit is very, very large. And just to show that this is not some super abstract concept, a little bit of basic physics for you. Just this kinematic equation, this trajectory there, that can be a simulator in itself. It gives us this little ball thrown in 2D. And so this would be sort of using the gradients to fit parameters of the simulator, which in this case is sort of initial position, velocity, and acceleration. So yeah, so this is kind of a trivial example, but of course you can get arbitrarily complicated. And the way that we do fitting with this is very similar to the analysis by synthesis approach before. And this is just instead of adjusting parameters of your scene and adjusting parameters of your neural network, adjusting parameters within your simulation. And we did this in this rendering context, so just showing some positions on a sensor from each of the mirrors there, just showing that we can fit things around with this differentiable ray tracing. We also, not necessarily spatial signals, but just another example of this, we did a gradient-based calibration of a neutrino detector. So just showing a variety of 6-dimensional fits in kind of the physical model space. And so just kind of one last related thing to this is that I mentioned we'd have this 2D to 3D, which we mostly solved. And then we mentioned that we are doing this in order to connect back to the underlying physics. And the way that we are thinking about doing that is, again, with differentiable simulation. But in this case, it's more along the lines of fitting a signal component as opposed to fitting a parameter of a detector. And so this is just not going into any details here, but just an example of a fit of a particular component of signals within atom interferometry. And yeah, and so yeah, kind of have these 3 broad questions, and really the methods are the answers to those. So 3D structure from 2D data we do with multi-view imaging and analysis by synthesis, and again using these tools of neural fields and ray tracing. For representation and calibration of our detectors, neural fields and differentiable simulation are also powerful, and then fitting components of our signal of our data is differentiable simulation. So yeah, thanks very much, and yeah, curious for any discussion.
Thanks, Sean, for that really interesting talk. If anyone has any questions, can please walk up to the mic. I wanted to start with one. So we've talked a little bit in the workshop about the challenges of 3D imaging and how much easier 2D imaging is. What do you think are the potential applications of something like radiance fields to taking 2D data and inferring something about 3D, and how much depends on like the specific camera setup you have?
Yeah, so I think like in our context, then the reason why we kind of care about it is that like what we're doing in the end is connecting a simulator that's happening in 3D to something measured, and so it kind of bridges the gap. Yeah, I think in general, like there are, of course, depending on your imaging, depending on the amount of information that you're able to capture with whatever constraints, then you can have artifacts, and that can be maybe not the best. That can distort your physics, or biology, as the case may be. Yeah, I think we're still, I guess, exploring a bit whether this, in our context, like doing this 3D reconstruction actually helps, and I think it'll be interesting once we kind of get closer to trying to apply this in a real extract physics information setup. But yeah, I think it's like kind of the two, balancing the 2 aspects of like, okay, reconstruction is a whole separate beast, but also like where things are actually happening is in 3D, and so yeah, trying to figure out some good in between for that.
Yeah, cool talk. So I've got 2 questions. So I was kind of curious, but maybe it's kind of like a straightforward answer, but how do you go from comparing your simulated, 3D model back to the original? Do you take it back to those like your original detector fields of view of the simulation and then do a difference there, or what does that difference function look like?
Yeah, so the difference is on the camera images. So yeah, this will work. But so the yeah, so you do the rendering of your what's being seen in your camera or in your optical setup, and then you do a pixel-wise comparison.
Right. Okay.
Yeah.
And so then I was curious. So when you go back to your like atomic views, like presumably, I don't know, with the integration time for detector, there's some uncertainty in the position. And I was kind of curious, how do you factor that uncertainty into like your simulated variance or stochasticity back to the image?
Yeah, so I think, yeah, I guess a couple of things. I mean, like, I guess in terms of like a stochasticity, like on like detector noise or what have you, lots of this is based around mostly like point estimates. And so essentially kind of finding a mean structure or whatever. It would be, I think, an interesting extension to do a distributional something or other, which having a differentiable simulation or having these nice representations like is nice for. I think for like uncertainties on neural fields is a research topic, and one that's very interesting, I think, but yeah, but it's a research topic. Yeah, in terms of some of the like uncertainties, like on the side of like detector setups and whatever, what we're looking at now is to try to do a software calibration with these differentiable simulations to try to minimize differences between simulation and data. Then yeah, of course that may also not be perfect, but yeah, again, I think lots of interesting future directions in terms of putting some error bars on some of these results.
Okay, thank you.
Yeah, thank you.
All right, let's thank Sean for a great talk.
Yeah, thank you.
All right, and next up in our session, we have Akshay, who's going to tell us about taking deep learning and applying it to radiology.
Great, thanks for the introduction, and thanks for the organizers for this invitation to give this talk. I think for the next 10-15 minutes or so, I'll just be talking about some of the work that we're trying to do in the domain of radiology to try to solve a lot of computer vision type problems. And just as context, radiology involves a lot of X-ray data, a lot of MR scans, a lot of CT scans, and a lot of the tasks that we really care about are about trying to classify the images. So what sort of disease might be present in a radiological image? Maybe we care about things like segmentation, so trying to really understand what pixel level information there is. And I think more recently, there's a lot of interest in prognostication, which basically means that given an image, what will happen to the patient 5 years from now? And in this particular presentation, I'll be focusing on some new data-driven methods that actually have a lower reliance on large training data sets, which is a pretty practical problem that we have to worry about. And I'll be focusing more on the conclusions for most of these studies rather than the methods, but always happy to chat more about the methods. So, all right, let's try to advance, maybe. Oh, perfect, there we go. Okay, so here are some disclosures. None that I really think that affect the content of the presentation, except the fact that I'm a PhD researcher, not a radiologist, and I work in a radiology department, and I'm not a biologist either. So probably take everything I say with a grain of salt. So maybe as just a quick show of hands, how many people here have heard of a term similar to data as the new oil when we're thinking about deep learning systems? So I see a few hands going up, and this is generally a common sentiment. Whenever we want to train a novel deep learning model, whenever we want to solve a new task using some of these data-driven methods, the first thing that we typically think about is what sort of data sets do we have? How representative are these data sets? How well would my models necessarily generalize? And these concepts and questions have been at the forefront of a lot of general machine learning research because a lot of work that we do is in this notion of supervised learning. So what that really means is, let's say if I'm trying to solve some sort of image classification task, I might have 9 images in my training data set. And for each of these images, I can associate a particular label. So I have this really nice paired data set where for every input, I have some sort of output that I'm trying to learn a model for. So once we have these, we can put this all together in a big pot, stir it up a little bit, and then we'll come up with the model at the end of the day. But this really requires a reliance on having these paired data sets. So I think any ML talk is incomplete without pictures of dogs and cats. So whenever you're trying to train a classification model, your input data might look like an image and a corresponding label. You might have another data set with more images and the labels, and this goes on. And you can really try to build good supervised learning models in this manner. And probably 95% of all papers that we see, at least in the radiology domain, has this nice supervised learning paradigm. But a major challenge that exists is a lot of times we don't necessarily have access to these labels because generating these labels requires some sort of expert consensus from a radiologist. This can be expensive, this can be time consuming, and in a lot of cases, it's just not feasible to generate labels at the scale of the data that we have. So what do we do when we don't have these labels and we just have all these raw inputs? So this is a new field of research that's really emerging over the last 3 or 4 years. And that's known as this notion of self-supervised learning, where you can actually work without any particular labels. So let's say I'll have the same images that I'm starting off with. Instead of having some sort of labels about, is this a cat? Is this a dog? I can just create some arbitrary pseudo labels that leverages the innate properties of the imaging data itself. And I'll just train a model to learn these pseudo labels. And then once I have these pseudo labels, I can just take that model and then I can fine tune for the label data sets that I have. So what this really enables is we can learn from a large unlabeled data set. And then even if we have a few handful of labeled examples that I care about for my task of interest, I can still build good models. So now, obviously, the question that might be in your mind is, what are these pseudo labels? How can we generate good quality pseudo labels? And this is basically where you can get as creative as you want. And I'll just try to give a few examples of what people in the field are doing. So 5 or 6 years ago, one of these methods was one of the most promising methods where you can take an arbitrary image and then you just rotate it. You can either keep it the same, you can rotate it by 90 degrees, 180 degrees, or 270 degrees. And all you train a neural network to do is predict one of those 4 rotation angles. And one might ask, well, why is this really important? Well, in order for us to be able to predict this rotation angle, we need to learn something innate about the characteristics of the image itself. So if we look at the second row image, it's unlikely that we might see this particular image out in the real world where a bird is hanging upside down on the tree branch. The third image is probably even more unlikely that the bird would be upside down. So based on the innate properties of this image itself, we start learning some sort of semantic relationships about the natural world in and of itself. So you can actually pre-train a model just by trying to learn some of these rotation tasks. And then you take 50 or 100 labeled examples, and you can actually build pretty robust models using that limited training data. Another pseudo-labeled training technique that I quite like is this notion of patching. So we can take an arbitrary image, and then we can divide it into these 9 patches. And then during training time, we'll just sample the center patch, and then one of these other 8 patches. And then the goal of the neural network is try to learn what are the spatial relationships between these 2 random patches. So my blue image might be my key, and then my red might be my query. So I just have to learn relative to this blue image, where does my red image go? And for me to understand this locationing, I don't really need to know that I'm looking at a cat, but innately, to solve this particular task in a supervised manner, I need to know that ears generally are pointed. Ears usually go above the eyes. So you're building up this really nice semantic understanding of the domain that you care about without explicitly using labels for your downstream task. And then once I've trained this model, all I really need to train the model is to call these sets of features a cat. And a different sets of features might be a dog, or it could be some sort of disorder on a radiological image. So I think this notion can be pretty powerful, where we're just learning in this paired manner, you're trying to predict the specific locations. So these are a couple of pre-training tasks that have been working quite well for computer vision. More recently, I don't think we can talk about machine learning without talking about natural language processing. So if we go back a few years and think about these bird style models, these actually work in a similar self-supervised setup. So you can take any arbitrary sentence. So today is a blank day in Palo Alto. And what your neural network will do is it will take these sentences, mask out or just remove a random word in there. And then all the goal of this neural network is to try to predict what word should go in this mask. And then the output is basically a probability distribution function. So if you really want to predict what should go in that particular mask, maybe because there's a lot of content about Palo Alto and weather that says that Palo Alto is either foggy, beautiful, or sunny, these are the top choices that your model has to learn. So again, based on this self-supervised task where we don't need any downstream labels, we learn something about the domain of the data that we're dealing with. And then these same concepts can be lifted into our particular domains of interest. So for example, we can take this image masking concept to some radiological images. So here we're looking at some MRI scans of the knee, where we're interested in trying to solve a segmentation problem. We really want to understand how much cartilage right here would this subject have. This is pretty painstaking. This was actually the exact reason 10 years ago I taught myself machine learning, because I refused to segment images by hand. So what you can really do is take this image and then randomly mask out parts of the image. Or we can take different image patches and then just swap their locations. And then the pretext task that my model is solving is just trying to recover the original image. And one can imagine that a model will learn to recover a good quality images if it learns something about the structure of the knee itself. So if it learns, that my patella has a particular appearance, it's always in front of my femur. So you're learning some domain-specific features. And then we can take this model and then we can adapt it for our downstream task of interest. And this provides us with a lot of benefits. So here on the x-axis, we're looking at a segmentation model that's been trained only in a supervised manner. And the y-axis is a segmentation model trained in the self-supervised manner with the same exact number of labels. We've just been able to pre-train our model with self-supervised learning. And then the dotted line is the unity line. So basically, any point that lies below this line tells you that the error that you get with your naive supervised learning methods is actually always higher than the error that you get with these self-supervised methods. And nearly every single point in our testing data set for every different patient that we're working with is below this y equals x line. So what this is really showing is that by doing this self-supervised learning, we're actually building up a lot of robustness in our models. In every single scenario, our data points are either the same or improved quality. So hopefully we're actually trying to get a bit of a free lunch here without necessarily using any new labels. And then the other benefit is that this really benefits scaling with large unlabeled data sets. So here we're working in different labeled data regimes. So we can have either 100% of our labeled data, 50%, and so on. And then on the x-axis, we're seeing what happens when we have larger and larger amounts of unlabeled data sets. And we see that as the extent of our pre-training data increases, we can see that our segmentation models get better and better over time. And some of these curves start saturating at some point, but if you're operating in a very data limited regime, so specifically this blue curve where we only have a handful of labeled training data, we can see that if we would have added more pre-training data, the model performance would have kept increasing more and more. So this masking-based approaches are one premise for self-supervised learning. Another popular approach is this notion of contrastive learning. And in contrastive learning, what we're doing is we're just playing a simple matching game. I'll take an arbitrary image, so in this case, it's a cat once again. And then what I'll force a neural network to do is I'll try to learn a neural network so that the way my network represents a cat is the same as its representation for another similar cat. And then if it should have different representations for all things that are not cats. So it's a simple matching game where we're forcing neural network features to be similar for similar concepts. And the way we can do this is we can take some arbitrary image and then we can keep transforming it or augmenting it in different manners. And then we can do this for all our images in our training data. So let's say if my training data has two images, a cat and an elephant, I can perform augmentations, and then I'll just train a neural network with the enforcement that the representations for my cats should be similar. The representations between the two elephants should also be similar, but the representations between these two cross-species pairs should always be different. So we're basically creating this partitioned neural network representation space, and then that's the pretest task that we care about. Then we can just take these methods and apply them to our domain-specific task of interest. One big area of research within this notion of contrastive learning is, well, a lot of the features that you'll end up learning really rely on what sort of augmentation schema that you have. So we really wanted to explore this for a lot of radiology applications. And we just wanted to understand, given the general homogeneous nature of radiology images, where the average chest X-ray across patients generally looks the same. It's only the subtle features that make a small difference. So we just wanted to see different augmentation methods. So these are the results, but at a very high level from this analysis, we learned that a lot of methods that work for natural images don't transfer for medical images, and simply cropping your images gives you the best quality representations for your augmentation schema. So this is really trying to take concepts that are somewhat popular in the general computer vision field and making them work nicely for our domain of interest. So we thought that this was pretty useful. And now whenever we run all of our contrastive learning methods, we don't even care about most of these other augmentations. We just randomly resize and crop, and that generally improves model performance consistently. So far, we've mostly been talking about vision-only models, but I think over the last year and a half, there's a lot of interest in text-to-image models, sure, this notion of multimodal learning. And maybe we've played around with models like DALL-E or Stable Diffusion, but they're very powerful and you can get very creative with it. So let's say if I just have a prompt of a photo of a corgi wearing a hat in Times Square, I can feed this into a neural network and it'll generate an image that looks really nice. This image doesn't exist in the world, but we can learn adequate representations to be able to synthesize this image. We can get even more creative with some of our prompts, majestic oil painting of a raccoon queen in a French setting. So we're really just limited by our creativity in terms of some of the objects that these models can represent. But the challenge once more here is that these don't necessarily represent domain-specific concepts that we might care about. So if I take the same model and if I prompt it with a picture of a lung X-ray, I'll get something that looks cartoonish. If I prompt it with a lung X-ray with a specific abnormality, again, this might go in a nice comic book or a kid's book, but if you show this to a radiologist, they'll laugh you out of a room. So something that we wanted to care about is how do we leverage some of these baseline models that have been trained with over 2 billion pairs of images and text, and how can we do domain-specific adaptation in a data-limited setting? And it turns out this is possible. I'll spare you the details for now, but we can really take some of these large-scale models and then doing some few-shot learning, we can actually adapt them to generate radiographs completely from scratch. So we can prompt our model by saying, generate an X-ray with a small right-sided pleural effusion, and we get something that looks like this. This model learns the difference between small and big. So these are the pleural effusion features that we're looking at. It learns the difference between right-sided and left-sided. It learns negation. So all these models have a lot of innate capabilities. We just need to fine-tune them in an appropriate manner to adapt them to our specific use cases. And it was interesting that we're able to do this across a wide range of abnormalities, completely in a self-supervised manner. We just took images that had presented to the hospital, we took the corresponding radiology reports, and then we trained them in a joint self-supervised multimodal manner. And we can keep prompting this model with different examples of imaging findings one might see in the clinic. So our goal here right now is if we can generate all these synthetic images, maybe we can train our next generation classifiers on the synthetic data itself to see whether or not this might alleviate the burden of not having labeled examples to work with. So those experiments are in process, and hopefully in a month or so, we'll have a bit more intuition of whether or not that works. So at a very high level, some takeaways from this talk is the fact that in biomedicine, we don't always have high quality and large labeled data sets, but just because we don't have labels doesn't mean that we don't have models. I think there's a lot of creative ways one can get around this notion just by having access to a lot of raw unlabeled data in the first place. I think one note of caution is that a lot of methods that might work nicely in papers in CVPR in the natural domain don't necessarily transfer directly to biomedical domains. So injecting some of our own domain knowledge and crafting and refining these methods is definitely a necessity. And then the last observation that we've had, for better or worse, is that scaling models in terms of how big the model size is and how much pre-training data you use generally consistently leads to better performance, which can really make us question, well, how does academia, which is more or less compute starved compared to the Googles and Metas of the world, what is the role of research in machine learning? But I think these are just some interesting observations that are coming out. If any of these methods might be interesting, we try to open source all of our code. We try to open source all the models. You can grab all of that information here, and I can go back to that slide later. But with that, I'd just like to thank a lot of the students in the lab who made all this work possible, as well as our funding sources, and happy to take any questions. Thank you.
Thanks, Akshay. That was really interesting. I think a lot of the issues you brought up of the CVPR papers not applying to radiology, we have equivalent issues in microscopy, especially I think because we often have very high dimensional imaging data. So even basic things like the shape of the model doesn't apply. Could you talk a little bit about what type of fine tuning you did that allowed you to get good predictions for your radiology images?
Yeah, it depends on what your domain of interest really is. I think one area that we're particularly excited about actually comes from the Stable Diffusion model, where you can take a natural image prompt and be able to use that to synthesize a medical image. The first part of that model is actually a variational autoencoder. So what that's learning is instead of processing your images in the image space, it condenses them into some sort of latent space. So we're actually using that quite successfully. So a lot of the mammograms, I didn't show any mammograms here, but a lot of the mammograms that we work with, they're high-res images, they're 4K by 4K. So maybe nothing like pathology or microscopy data, but that's high resolution for us. Instead of working and building models on those 4K by 4K images, we just pass it through this variational autoencoder, and we can get something between like a 16 to 25-fold image compression. And then all of our downstream models we build are just in the latent space of the VAE. Based on that, which we had pretty good observations with, we just decided to train our own medical imaging specific VAE that's been trained on 2 or 3 million medical images. And now that's our image compression method. So instead of working in a very high dimensional space, we're at least working in a bit more of a compact space. And we see that you don't lose any sort of downstream classification or segmentation accuracy. And you can build bigger models, or you can train faster. So you get a lot of computational benefits, and we're exploring that more and more.
Okay, questions from the chat. Compared to supervised learning, how much more data do you need for SSL?
It depends. Now, data can be split into 2 buckets. Data could be either labeled data or data could be unlabeled data. So the first observation is get as much unlabeled data as you can. Even if it's not exactly in your domain of interest, I think you can still learn interesting features based on what your pretext task is. And then when we're talking about labeled data, again, more is always better. But we've seen that when we self-supervise, where we have equal amounts of labeled and unlabeled data, we can actually get by with maybe 4x or 10x lower label data training needs. So for segmentation models, let's say if we trained a segmentation model with 100 patients, with self-supervised learning, we can actually train the same model with like 10 to 20 patients or so with the same accuracy metrics or the same robustness, probably with a little bit of increased robustness. So I would say at least you can reduce data requirements by 4-fold pretty well. But then depending on what sort of methods you're using, you can really push that forefront.
Great. Thanks for the interesting talk. How does your model perform on 3D imaging data?
Yeah, so all the examples that I showed here are 2D, but I think a lot of the real focuses that we have in the lab are for 3D approaches. So we work a lot with 3D MR scans, 3D CT scans. Same concepts apply. So a lot of the NEMR images that I showed you, those are just 2D slices from a 3D image stack. With a lot of the self-supervised learning that we've done, I think 3D presents a few more computational challenges, but the methods just scale linearly across dimensions. So if we think about NLP, that's a one-dimensional signal. If we're thinking of a masking task, we can simply extend that to 2 dimensions and we learn pretty robust representations. We've done the same for 3D patch-based masking. That seems to work well too. So I think we're taking a little bit of a step back saying, well, a lot of the 2D methods work pretty well. So can we just learn 2D representations and then lift them into 3D, and you can have some sort of temporal or spatial consistency that we can enforce. So that allows us to take some pre-trained models from the natural domain and then adapt them to our tasks. So I don't think for a lot of radiology applications, there's a huge need for domain-specific 3D models. I think you can get pretty far with 2D models too, which provide some computational benefits.
In radiology and pathology, the disease diagnosis might not be clear cut. How does this, like maybe not being able to have a ground truth at all, affect the supervised versus unsupervised trade-off?
Yeah, I think your models will only be as good as your training data and what sort of benchmarks you're evaluating. So that's another focus that we have is, can we create high quality benchmarks for the community where instead of just relying on things like ICD codes, which is quite common for a lot of domain hospital specific models, can we in fact get a panel of 3 radiologists and have them look at the same image, and you can't leave the room till you've reached some sort of consensus? Again, there's pros and cons to this multi-reader study, but I think it'll always be a limiting factor. But then on one front, these are the decisions that the radiologists are making already. So at least if we can be on par with the current radiologist accuracy metrics, maybe that's a good first step, because even when we build a model, that doesn't necessarily make it useful. We still have to think about, well, how do we deploy it in the hospital? How do we make decisions based on that model? So I'd rather not have the perfect model as a first pass, deploy it, learn something about it, and then come back and iterate on it. But I think the higher the quality of labels you get, the more human expertise you'll require. So it's a trade-off of, do you want 5 people labeling one image or 5 people labeling 5 separate images? And right now I'll take the latter.
Awesome. Why do you think cropping was the only augmentation that was applicable to your domain? And do you think that's generally true in microscopy and other areas, or is it something specific?
Yeah, that's a very good question. I don't think we have a very clear answer why cropping in particular works. I think it has something to do with the extent of the findings that we expect to observe. So maybe, let me see if it's feasible to go back. Yeah. So in this particular case, so we have a lot of pixels in this particular image. But if I'm trying to diagnose something like a little tiny nodule in the lungs, so the lung nodule might only be like a small area right here. So for a lot of these other methods like the blurring, noise addition, Sobel filtering, we actually end up losing that particular feature because of the augmentation. So we might have an original image with the abnormality, and then the augmented image loses that abnormality. In contrastive learning, we force both of those to have the same representation. So actually telling your model to be invariant to the pathology that you're trying to detect, that's one of our clues as to why cropping might work. Because even if we crop, we maintain the general structure of the image. The finding always persists in there, but that's a hypothesis. I think other people have made similar observations across different domains, and there's still no right answer as to why it works. But at least when we evaluate this on high quality label training data, it seems to always be robust. So we're trying not to lose too much sleep over it.
Awesome. Well, join me in thanking Akshay for a really great talk.
Thank you.
All right, we're going to take our break now, and we will return at 9:45 for our last session of the workshop.